Okay, hello to all of you. Let's discuss chapter number 9. And we go into the slideshow mode. So, in chapter number 9, we are going to talk about nucleic acids. Okay, so nucleic acids. Nucleic acids, basically, there are two kinds of nucleic acids. There's DNA and RNA. That's what we're going to talk about in this chapter. So, basically, nucleic acids. So let me give you some little background about nucleic acids. So basically, as I said, there are two kinds of nucleic acids. We're going to discuss about DNA and RNA. We're going to talk about the differences. We're going to talk a little bit about the structures. We're going to talk about different kinds of RNA, different conformations, different types of DNA also. And we're going to talk about how the information flows, from DNA to mRNA and from mRNA to proteins. So first of all, let me give you some background about this topic. So basically, as we know that if you look in a family, um, we can see some of the characters which we share with our parents or with our grandparents, or some characters, we have some similar characters with our siblings also, right? So there are some characters which run in the families. So how do these characters, they're being carried on in the families, or how do we inherit some of the traits from our parents? The traits or the characters include various things, which include our skin color or the texture of our hair, how do we behave, what kind of diseases we are susceptible to. So um, all these things basically are we are carrying in our genes, which we are getting from our parents. So basically, what our parents are giving us, they're giving us genes, right? And these genes, where are these genes? These genes are present on the chromosomes. So where are these chromosomes? These chromosomes are present in the nucleus of our cells. And these chromosomes, they have genes on them, right? And these genes, they are made of DNA, okay? And this DNA, which is a very, very long molecule, it's negatively charged. We will be talking about the structure of the DNA, and you will see that DNA is negatively charged because of a phosphate group in it. And this DNA is wrapped over some proteins. That's the way how this long molecule, which is carrying a lot of information, is stored in our cells. Our cells are very small, and nucleus is even smaller. So how it's all packed inside? Because this DNA long molecule, which is carrying a lot of information, is all wrapped over these proteins. So DNA is the code of life. It's the blueprint of life. It's the secret of life, right? Because all the information is there, okay? But how does this information actually functions? You know, DNA is basically a database, okay? It is a lot of information. It's a lot of knowledge. But this DNA is itself not performing the function. Who is performing the functions? Who is the real player? The real players are proteins. Proteins, we have already discussed in our first lecture what proteins are. We just discussed in chapter number 8 also. These proteins are present in the membranes, and they are doing majority of the functions. So the real players in our cells are proteins, because our cell, we are nothing but made of trillions of cells. So our cell is a small machinery, and how it is working? It is working all with the help of proteins. These proteins are very versatile. They are performing various functions. They can act as enzymes, which we discussed in lecture number, which was our topic of week number 2. So the majority of the enzymes are protein in nature. They can speed up the rate of reactions. They act as organic catalyst. These proteins, in chapter number 8, we just talked about their transport proteins. They help in the transport of various things across the membrane. Then these proteins can act as receptor proteins, which means they can receive signals from the environment, and they can lead to many changes in our cells. These proteins, they are involved in cell to cell recognition. They help in cells attaching to each other, because these cells, they adhere to each other, they connect to each other, and they form tissues. Then tissues form organs, organs form organ systems, and then organ systems form the organism. Like we are an organism, we are nothing but cells. So proteins are there in our cytoplasm also. They give cytoskeleton, they make the skeleton of our cell. They're doing many functions. So proteins are the real players, and the information, what kind of a protein will be made, that information is there in our genes, and our genes are made of DNA. And this information, to code proteins from DNA, it involves two steps. First step is called transcription, second step is called translation. In the first step, which is called transcription, the information which is there in DNA, which is there in our genes, is first of all converted into mRNA. mRNA is messenger RNA. Messenger RNA is one of the kinds of RNAs. We will discuss all those kinds in this chapter as we move forward in this chapter. And then the information which is there in the messenger RNA is going to be converted into proteins. So the second step is called translation. So this is our topic. Okay, first of all, we are going to talk about basically we are going to talk about the structure of DNA. Then we are going to talk about, in between we will be comparing it with RNA also, and we will talk about then different kinds of RNA, and then we are going to talk about how the information flows. Okay, so that's what we are going to learn in this chapter. So basically, similarly like the structure of protein, we can study the structure of nucleic acids. Also at basically the order of the nitrogenous bases, which I'm going to discuss what nitrogenous bases are, which I'm coming very shortly on that. Order of bases in the polynucleotide sequence. Before we go into further details of this chapter, I would like to remind you again that nucleic acids are also polymers. Okay, so there are 4 different kinds of biomolecules which we are going to learn in this course. We have already talked about proteins. In the last lecture we talked about lipids. Now we are talking about nucleic acids. This is the third biomolecule we are talking about. One of the very important characteristic of these biomolecules, the molecules of life, is that they are macromolecules, means they are big molecules, and they are polymers, which means they are made of repetitive subunits. They are made of repetitive monomers. Proteins, they are made of amino acids. We are going to talk about nucleic acids. Nucleic acids is also a polymer, and its monomer is a nucleotide. We talked about lipids also. Lipids are biomolecules, but remember that they are not polymers, which means they are not made of repetitive subunits. That's we discussed in the last lecture. And moving forward, we will be also talking about carbohydrates. You will see that the carbohydrates are also made of simple sugars. That's the monomer of the carbohydrates, and they're called monosaccharides. So let's come back to our topic, which is the nucleic acids. So important thing to remember here is nucleic acids, they are polymers, and the monomer of a nucleic acid is called a nucleotide. And I'm going to talk about the structure of the nucleotide. In that nucleotide, we have some nitrogenous bases, and the order of these nitrogenous bases makes the primary structure of the nucleic acids. And what is the secondary structure? It's basically the conformation of the this is basically the conformation of the nucleic acid strands is basically the secondary structure. And then tertiary structure is then these molecules, they can also supercoil over each other. That is called its tertiary structure. And quaternary structure is when these nucleic acids, they interact with some other macromolecules like proteins. So there is some information about this structure of nucleic acids at 4 different levels is also here. Primary structure, sequence of bases. Secondary structure is the ordered arrangement of nucleic acid strands. That is the secondary structure. Then the tertiary structure is the supercoiling, and the quaternary structure is the interaction of the nucleic acids with some other macromolecules like proteins. Okay, let's first of all talk about what a nucleotide is. Nucleotide is a monomer of nucleic acids. Okay, and let me show you. I have to go to this slide to show you something. Yes, this is the structure of the nucleotide. Let me zoom it a little bit. See if I can zoom this. Okay. Okay. So you can see here. Now, if you see on the left side, it's written ribonucleotides, and on the right side, it's written deoxyribonucleotides. What does ribonucleotides mean? As I just said, there are two kinds of nucleic acids. There is RNA, there is the DNA. RNA is called ribonucleic acid and DNA is called deoxyribonucleic acid. Now, the nucleotides which are present in RNA, they are called ribonucleotides. And the nucleotides which are present in DNA, they are called deoxyribonucleotides. We are going to discuss very shortly why they are called deoxyribonucleotides and why they are called ribonucleotides. Okay, so this is the structure of a nucleotide. Every nucleotide has this pentose sugar. Okay, pentose sugar means a sugar which is carbons. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, and on carbon number 4 is dash carbon number 5. Okay, this is a pentose sugar. On carbon number 1 is attached the nitrogenous base, and on carbon number 5 is attached the phosphate group. This is a structure of a nucleotide. A nucleotide has three components: pentose sugar, nitrogenous base, and phosphate group. Now, there is a difference in the pentose sugar which is present in RNA and the pentose sugar which is present in the DNA. Now, in the DNA, if you look here, at carbon number 2, which is present is hydrogen, and in RNA, the group which is present is OH. So, RNA has one extra oxygen. DNA has only hydrogen, RNA has OH, which means it has one extra oxygen. So, there is one oxygen less in DNA. There is one oxygen less in DNA. That is why it is called deoxyribonucleic acid. And RNA has this one extra oxygen. It has oxygen. That is why it's called ribonucleic acid. And the sugar, sorry, I forgot to tell you this. The sugar which is present, this pentose sugar which is present in RNA is called ribose sugar. And the sugar which is present in DNA, is called deoxyribose sugar. Okay, this sugar is called deoxyribose sugar. This sugar is called ribose sugar. That is why DNA is called deoxyribonucleic acid and RNA is called ribonucleic acid on the basis of the kind of carbons they have in them. Okay, so this is the basic structure of nucleotide I wanted to show you. Let's go back and let me show you again. It's very clearly shown here. The sugars, this is the sugar in RNA. This is the sugar in DNA. You can see at carbon number 2, there is H here and there is OH here. So this is ribose sugar. This is deoxyribose sugar. Okay. So basically, we discussed nucleotides, three components: pentose sugar, nitrogenous base, and the phosphate group. Now these nitrogenous bases, they are called nitrogenous bases because these are ringed structures in which there is carbon as well as nitrogen. Because they have nitrogen in them, they are called nitrogenous bases. And why they are called base? Because if you put them into water, the pH will be alkaline, so that's why called the bases. Now two of nitrogenous bases, they are called purines and they are called pyrimidines. Now purines, they are two ring structures, and pyrimidines, they are single ring structures. So the purines have two rings. One ring is 6-membered, another ring is is a 5-membered ring. And the pyrimidines, they have one 6-membered ring. Okay. Now purines, they have two nitrogenous bases called adenine and guanine, and pyrimidines, they have cytosine, thymine, and uracil. So these are purines. You can see they are double ring structures. This is a ring with 6 carbons, 6 members, and this is a ring with 5 members. And in the pyrimidines, there is one, they are single ringed, they have 6 members. So in purines, we have two nitrogenous bases which include adenine and guanine. So adenine and guanine, they are both present in DNA and RNA. In the pyrimidines, there is a cytosine and there is thymine. Cytosine and thymine. So cytosine is present both in DNA and RNA. Very important thing to remember here is thymine is mostly present in DNA. Okay, it's mostly present in DNA, but it is not much in RNA. Okay, so it is mostly present in DNA. In RNA, instead of thymine is uracil. Okay, so thymine is mostly present in DNA and uracil is present in RNA. So this is these are the two different categories of nitrogenous bases. Okay, now something important. So we just discussed what nucleotide is, which I showed you here. This is the structure of nucleotide. Now there is one more term which you need to know. What is a nucleoside? Nucleoside is the pentose sugar when it is attached to the nitrogenous base. So pentose sugar attached to nitrogenous base is called nucleoside. And if phosphate is attached here to the fifth carbon, it becomes a nucleotide. So nucleotide is without phosphate, so it lacks phosphate group. So nucleoside has pentose sugar and the nitrogenous base, and if the phosphate group is also attached, it becomes a nucleotide. So we discussed about nucleotide, nucleoside, nitrogenous bases, and these are different kinds of nucleotides. Okay, so you can just see them. Now next question is, as I said, nucleotide is a monomer of nucleic acids. So now nucleic acids, they're polymers. So in these polymers, all these monomers, they're connected to each other by covalent bonds, and the formation of covalent bonds in all the biomolecules is by reactions called dehydration reactions, or they call condensation reactions, which means that two monomers together lose a molecule of water, and that links these two monomers by the formation of a covalent bonds. So let's talk about how these nucleotides, they are linked in DNA and RNA. So I'm going to skip all this because I'm going to explain through this picture. So I'm going to come here and I'm going to explain you this. So this is RNA and this is DNA, right? Because here is OH. So we can, if you have to recognize if this is a strand of DNA or it's RNA, you have to go and look on carbon number 2. If it is the OH group, it is RNA. If it is H group, it is DNA. So this is DNA, this is RNA. So there are three important differences between DNA and RNA. Number one, DNA is double-stranded. DNA has two strands. RNA is single-stranded. In DNA, the sugar is deoxyribose sugar, and in RNA, the sugar is ribose sugar. In DNA, there is thymine. Mostly thymine is present in DNA, and RNA has uracil instead of thymine. These are the three main differences between DNA and RNA. Now, the question we need to now understand is how these nucleotides, they're connected to each other so that DNA forms a big polymer. So this is, you can see this is first nucleotide, ribonucleotide, this second ribonucleotide, third and fourth. Now, how they are going to be linked with each other. So we know that it's an important property of all these biomolecules that they're all connected to each other by covalent bonds. And these covalent bonds, they have different names in different biomolecules. The name of covalent bonds in proteins is peptide bonds. So now we're going to talk about what is the name of the bonds in the nucleic acids. The name is phosphodiester bonds. Phosphodiester bonds, and how they are formed. They are formed by, again, by dehydration reactions. So you can see here, this is first nucleotide, this is second nucleotide. On carbon number 3, okay, on carbon number 3, in every nucleotide there is a OH group. Let's go back here and see. You can see here. So I'm not talking about carbon number 2, I'm talking about carbon number 3. In every nucleotide at carbon number 3 there is a OH group. You can see in all these there is a OH group. Okay. So basically there is a OH group here, and in the second nucleotide there is a phosphate group here, which is attached to this phosphate group is attached to the carbon number 5 of the second nucleotide. Okay. So on carbon number 3 of OH, on carbon number 3 of this nucleotide is the OH group, and on carbon number 5 is attached the phosphate group. Now this OH group, which is present here on the 3' end, and the phosphate group which is present, on the fifth carbon of the second nucleotide, they together lose a molecule of reaction, which is a dehydration reaction, and that results in the formation of a covalent bond, and that's called a phosphodiester bond. Okay, that bond links the two nucleotides. Okay, so you can see here this phosphate which is attached to this carbon here on the carbon number 5, this is linked to this nucleotide. So this phosphate is attached to this carbon, as well as it is attached to this carbon which is present here of this nucleotide. This is the carbon number 3 of this nucleotide, and this is the carbon number 5 of this nucleotide, and this phosphate is attached to these two nucleotides. So this kind of a linkage is called a phosphodiester linkage. So this is the way. So through this phosphate group which is attached to this 3' of this nucleotide and 5' of this nucleotide, these two nucleotides, they are linked to each other and they're called phosphodiester bonds. So this is basically shown. So this is a strand of RNA, and in this you can see this is your pentose ribose sugar, which is a pentose sugar. On carbon number 1 is attached adenine. Here the cytosine is attached. Here guanine is attached. Here uracil is attached. Okay, and phosphodiester linkages, they basically form the backbone of DNA and RNA. So same thing is here. So you can see here, this is the deoxyribonucleotide, because this is the DNA strand. So this is H here, so we can recognize this is DNA strand. So here is thymine, here is guanine, cytosine and adenine. Okay, and you can see here that this is the phosphate group which is attached to this carbon of this nucleotide. So this carbon belongs to this nucleotide, and this is the OH group which belongs to this nucleotide. Okay, so this phosphate is first of all linked with this carbon, as well as it is linked with this carbon which is through the OH group which is attached to the 3' carbon of this nucleotide. So this is called phosphodiester. This is called the phosphodiester bond. So this is the way how the nucleotides, they are linked with each other. Okay, I think now we are ready to understand the structure of DNA. So as I said, DNA is double stranded, which means there are two strands of DNA which are linked to each other. So you can see here, this is one strand of DNA. Okay, when I talk about the strand, I am talking about this. Okay, so RNA is single stranded, but DNA is double stranded. So this is the structure of DNA. There are two strands. This is one strand going around like this, and this is the second strand of DNA, and they are wrapping around each other. We call them, they are wound around each other, and they form the double helix. Some of the important things to know here are, when the two strands of DNA, they are wrapping around each other, or they are wound around each other, the two strands of DNA, they are linked with each other, and they are linked with each other by hydrogen bonds, which are very weak bonds, and they are linked to each other by complementary base pairing rules, according to which A always links with T, C always links with G. Okay, so you will see that this is A nitrogenous base which belongs to this strand. This is thymine which belongs to this strand. This is cytosine which belongs to this strand. This is guanine which belongs to this strand. So A always links with T, C always links with G. So A is linking with T through 2 hydrogen bonds, and C is linking with G through 3 hydrogen bonds. So it's very clearly shown here. This is the structure of adenine, this is the structure of thymine, and you can see that the hydrogen of this is forming one hydrogen bond with this oxygen, and this hydrogen is forming second hydrogen bond with this nitrogen. So these are 2 hydrogen bonds, and this is cytosine and guanine, they are forming 3 hydrogen bonds between each other. So the two strands of DNA, they are linked to each other by hydrogen bonds, and they're linked to each other by complementary base pairing rules. Another important thing to know about the double helix structure of DNA is that the two strands of DNA, they are antiparallel in nature. So in the DNA, if this side is 3 prime, the opposite side is going to be 5 prime. So so what does 3 prime and 5 prime basically means? 3 prime, so 5 prime means, so this is a nucleotide, this is nucleotide. So this side is this is the carbon number 1, 2, 3, 4, and on the is attached the fifth carbon, this is, and on the fifth carbon is attached the phosphate group. So you can see the orientation of this strand, that in this strand the orientation of phosphates is on this side, which means that, so phosphate is attached to the fifth carbon, so which means that in this strand of RNA or this strand of DNA, all the phosphates are on this side, which means 5 prime is on this side. Okay, so which means if the phosphate is on this side, the hydroxyl group is on this side. So it means that this is 5 prime, so opposite side is 3 prime, which means in this strand of DNA, all the phosphates are facing this side and all the 3 prime hydroxyls are facing downside. So that is why this side is called 5 prime and this side is called 3 prime. But when we talk about the double stranded structure of DNA, the two strands of DNA, they are anti-parallel in nature. Basically, if this is the 5 prime in this strand, in the opposite strand there will be 3 prime. Actually what it is, the orientation of the pentose sugar is actually flipped in the second strand, and as a result of that, in the second strand all the hydroxyls are on this side and all the phosphate groups are on this side. So that is why the two strands of DNA, they are anti-parallel in nature. So this is one strand, this is the 3 prime, this is the 5 prime, and in the opposite strand, if this is the 3 prime, in the opposite strand it is going to be 5 prime. If this is 5 prime, opposite strand is going to be 3 prime. So these are two important things to know about the double stranded structure, and these, as I said, these two strands are wound around each other, and the way they are winding around each other, there is the formation of two grooves. One of the groove is called large groove. So this is the large groove. So you can see how they are wrapping over each other. This groove is called large groove, and this groove is called the small groove. And this is around 1 turn of the DNA, and there are around 10 nitrogenous bases in 1 turn. Okay, this is all what I just said is written here, so I'm going to skip it. This also we just discussed. Okay, there are different conformations of DNA or different kinds of DNA, B type, A type, and Z type. So basically, normally the DNA which is mostly present is of B type. It is having right-handed helix, and its diameter is 11 Armstrongs, and it has 10 base pairs per turn of the helix. ADNA is also right-handed helix, but it is thicker than BDNA. It has around 11 base pairs per turn of the helix. And ZDNA, it is left-handed DNA. It's a left-handed double helix. So here they are shown. This is A type, B type, and Z type. So this is left-handed. So these both are right-handed. This is left-handed double helix, and it plays a role in gene expression. And it usually consists of alternating purines and pyrimidine bases. Okay, so we just talked about DNA. Now we're going to talk about RNA. Some of the differences between DNA and RNA I have already discussed. This is all what is written here. I have already discussed the differences. This slide actually shows how the information flows from DNA to RNA to the proteins. As I've already given you background, that it's a DNA is a knowledge, it's a database, it's a information, it's a blueprint of life, it's a code of life, it's a secret of life, but the actual players are proteins. So this slide basically talks about how the information from DNA is converted into mRNA, and then it is converted into proteins. Before we go into the details of this, I think it will be good to talk about different kinds of RNAs first. Okay, so because some of the RNAs we are going to discuss, they will be, those terms will be used when we are going to discuss about how the information flows. So first of all, we have there are different kinds of RNAs. First of all, transfer RNA, we also call it tRNA. The function of transfer RNA is to transfer. So what does this transfer RNA transfers? This transfer RNA basically transfers the amino acids. So it recognizes a code and it codes for the amino acids. I'm going to show you the structure of that. What is sorry, ribosomal ribosomal RNA. Ribosomal RNA is a RNA which is actually a component of ribosomes. So what are ribosomes? Ribosomes are protein factories on which the proteins are formed. So ribosomes, they are some complex structures which are made of RNA, which is the rRNA, ribosomal RNA and proteins. So they are complexes which are made of rRNA and proteins. So ribosomal RNA is a component of ribosomes. Messenger RNA. Messenger RNA is a messenger of DNA. So all our genes which are made of DNA, when they have to express themselves, which means when they have to code for the information, when they have to code for the proteins, this process takes place with the help of the RNA called messenger RNA. So messenger RNA is the messenger of DNA. Then we have small nuclear RNA. This is a kind of a RNA which is actually helping in a very important process which takes place in eukaryotic cells. It helps in the processing of RNA. So it basically helps in removing some parts of the mRNA which are called introns, and it helps in the splicing of the axons together. So we will discuss about that also. Small interfering RNA and microRNAs, they are basically involved in some kind of gene expression, and these small interfering RNAs, these are basically used a lot by the scientists these days in the labs to knock out some of the genes. And long non-coding RNA, not much is still known about them, so they're still under debate. Okay, let's go talk about what a transfer RNA is. Transfer RNA, as I said, is that RNA which is responsible for transferring the amino acid. So it's a RNA, so it's a single stranded. So its structure is like this. So it's all single stranded because of the complementary base pairs present between a single strand. There's a possibility of formation of these loops. Okay, but remember that these are not two strands. This is just the one strand which is just looping. This looping is because of, like, if there is A here, it's if it is C here, it's G here, it's going to connect. They are going to link with each other because of the, if suppose here there is some complementation, then the RNA strand is going to loop like this. Okay, but this is single stranded. In the transfer RNA, on the 3' end, there is a amino acid binding site. Here the amino acid binds, and here is a code which is called anticodon, and it actually recognizes a code on the mRNA, and it brings the specific amino acid. That's the function of the transfer RNA. Ribosomal RNA, as I said, it's a component of the ribosomes, and ribosomes are the protein factories, and they are made of proteins and the rRNA. And in the prokaryotic organisms and eukaryotic organisms, they both they're different ribosomes in the prokaryotic and the eukaryotic organisms. I hope you know what prokaryotic and eukaryotic organisms are. Prokaryotic organisms are those organisms which do not have nucleus, and they don't have the membrane enclosed organelles like bacteria. They are prokaryotes. Eukaryotic organisms are the ones which have nucleus in them and the other membrane enclosed organs in them. Example is like animal cells, plant cells, they're all eukaryotic cells. So the ribosomes are different in prokaryotes and eukaryotes, but they are basically made of two subunits. One is a large subunit, another is a small subunit. Messenger RNA, as I said, messenger RNA is basically the messenger of DNA. In the eukaryotes, when the mRNA is formed, when it is first formed, it is not the mature form of mRNA. It has to be further modified, and then that mRNA is sent to the nucleus. Then that mRNA is sent to the cytoplasm to go and bind to the ribosomes, and then the protein is made. So messenger mRNA is the messenger of DNA. It is basically carrying the information from DNA, but the processing of mRNA is required in the eukaryotes, and there is no processing of mRNA required in the prokaryotes. Then the small nuclear RNA, this is also called snRNA. This is basically involved in the processing which I just talked about in eukaryotes is the processing of the mRNA takes place. So this kind of a RNA can complex with some proteins and they form small nuclear ribonucleoprotein particles. They are called snRNPs. They together basically helps in the process. In that process, all the introns, which we call a junk DNA, which does not code for the proteins, is all removed, and the axons which are coding for the proteins, they are joined together. That process is called splicing, um, and this snRNA complexes with some proteins and form snRNPs, and they help in that process. So this is about the different kinds of RNAs. Now I think we have enough information and we are ready to understand how the information flows. Okay, so this is the DNA. Okay, so now I'm just going to discuss with you how the information flows. So hold on. Okay, we have discussed all these things. Okay, so first of all, this slide is showing you DNA replication. First of all, there is another process going on which we'll be discussing in the upcoming lectures, DNA replication. So DNA needs to make its copies because this DNA is present in our cells, and our cells are dividing for the dividing loop. Keep on making the copies of itself. That process is called replication. Then the DNA, first of all, in the process of gene expression or protein expression, first of all, the information in DNA is converted into mRNA, which is the messenger RNA, by a process called transcription. And in this process, a very important enzyme called RNA polymerase is involved. So there are two strands of DNA. One of the strand acts as a template strand, and on the basis of whatever the sequence is there on the DNA strand, an RNA strand is going to be made. And then that mRNA is going to leave the nucleus and it is going to go and bind on the ribosomes. And on the ribosomes, the mRNA is this ribosome is to move on. This ribosome is actually going to walk on mRNA. There is transfer RNAs. So they're going to recognize a code on the mRNA, and that code on the mRNA is called codon. And that codon is recognized by a code on the transfer RNA. I show you here. Here is and that code which is present on the transfer RNA, it's called anticodon. And so the codon is present on mRNA, anticodon is present on the transfer RNA. So that is this transfer RNA is going to recognize code and it is going to bring the specific amino acids. And that's the way how peptide chain is going to be made on the ribosomes. We'll be talking about in the next week, we're going to talk about these processes in much more detail. So right now is just an overview, and you know that this peptide chain can fold further. We have talked about all these things and can form the three-dimensional structure of the protein. So this is the way how the information flows. Important thing to know about the differences in the transcription process which takes place in prokaryotic cells and the eukaryotic cells is, in the eukaryotic cells, the processing of the mRNA is required. So the mRNA which is from DNA is not a mature form of mRNA. First of all, it's called just a primary transcript. Then some processing takes place, and then it forms a mature form of mRNA, and that mature form of mRNA is going to leave the nucleus, and that is going to go to the cytoplasm and is going to go and bind to the ribosome, and then the second step of translation will take place there. So you can see here what are the processings required. So as I said, introns in the DNA are some regions which we call them junk DNA, which means they do not code for any protein. So these are axons. So all these dark green parts you see, these are axons. So all these in the processing part which takes place in eukaryotes, all these introns they are removed, and all these axons they are joined together, and that form a protein. And also the two ends of mRNA, they are also modified. So on the 3' end is attached a poly A tail, and on the 5' end is also attached the on the 5' is attached some modified nitrogenous base is attached there. So this is basically to modify the two ends of the DNA, of the mRNA. So this is all part of the processings which takes place. And this is what is being shown here in the prokaryotes. In the prokaryotes, you can see this is the DNA, these are the genes. So you can see that the mRNA is this is the DNA segment, and this is the mRNA which is being formed. And this mRNA does not need any processing. This mRNA, which once it's formed, it is ready. So this is the mRNA which is formed from this DNA, and you can see that this is the ribosome, which is binding here, and this ribosome is moving on this mRNA, and it is making the polypeptide. So this is this is moving on this and forming a polypeptide, and it reaches here. You can see this is gene A, this is gene B, this is gene C. So once the ribosome reaches here, this protein A, it will be the polypeptide chain will be made. It will further fold if the protein is ready. So this is the mRNA which is coding for gene B. So it's going to make this polypeptide B, and this part is going to make polypeptide C. So the important thing to know from here is in the prokaryotes, mRNA, once it is formed, it's ready for the next. It is ready for the next step, which is translation. There is no processing required in the prokaryotic transcription. The mRNA, once it's formed, it's ready for translation. No further processing is required. But in the eukaryotes, once the mRNA is formed, it needs some processings. It needs the removal of the introns. Its 2 ends needs to be modified. Only then it forms the mature form of mRNA. Only then it is going to go to the to the cytoplasm. It's going to go to the bind to the ribosomes. Only then the protein is going to be made. So that's all in this chapter. So that finishes this chapter. I hope you guys understand. If you have any question, please let me know.